For most of the last century, biologists assumed human evolution had effectively stopped. The idea was simple. Once our ancestors built shelter, learned to cook, and organized into societies, the brutal pressures that shape a wild species seemed to lift. We had, in a sense, outrun natural selection. A study published in the journal Nature on April 15th, 2026 shows that assumption was wrong. By reading the DNA of nearly 16,000 people who lived across the last 10,000 years, a team of Harvard geneticists found that natural selection never stopped working on the human body. In recent millennia, it actually sped up. To understand why this is a genuine surprise, you have to know what the field believed before. The specific force at the center of this study is called directional selection. It happens when one version of a gene gives such a clear advantage that the people carrying it have more surviving children, and that version spreads through a population. The textbook example is the ability to digest milk into adulthood, which became common in Europe only after people started keeping dairy herds. That example carries a puzzle of its own. People in Europe were milking animals for thousands of years before the gene that lets adults digest milk became widespread, which means the dairy farms came first, and the biology caught up only much later. Recent research suggests the gene spread fastest, not simply because milk was available, but during stretches of famine and disease, when the calories and clean fluid in milk could mean the difference between living and dying. Selection, in other words, often waits for a crisis. Until now, scanning ancient genomes had turned up only about 21 clear cases of this kind of selection in our species. 21. That small number led most researchers to conclude that strong, fast natural selection had been rare ever since modern humans first appeared in Africa around 300,000 years ago. There were two reasons the count stayed so low, and neither had much to do with the truth. The first problem was simply a shortage of data. To catch a gene variant rising or falling in frequency over the centuries, you need DNA from thousands of individuals spread across thousands of years. That kind of collection did not exist. The second problem was harder. When a gene becomes more common in a population, natural selection is only one possible cause. People migrate and bring new genes with them. Populations mix, and in small communities, gene frequencies simply drift up and down by chance, like a coin that lands heads 10 times in a row. Telling real selection apart from migration, mixing, and random luck was a problem nobody had solved. And nowhere was that problem worse than in West Eurasia. Earlier ancient DNA work, much of it from Reich's own lab, had shown that the people of Europe are not a single continuous lineage, but a blend of at least 3 very different populations. First came the hunter-gatherers who lived there after the Ice Age. Then, beginning around 9,000 years ago, farmers arrived from Anatolia, in what is now Turkey, bringing crops, livestock, and their own genes. Finally, roughly 5,000 years ago, herders swept in from the steppe grasslands north of the Black Sea, reshaping the ancestry of much of the continent. Each of those waves changed which genes were common without any selection at all. A region rebuilt 3 times over by migration is the hardest possible place to spot the quiet hand of natural selection, which is exactly why it had stayed hidden. The solution came from the laboratory of David Reich, a geneticist at Harvard Medical School and the Harvard Faculty of Arts and Sciences. Reich's lab spent 7 years assembling the missing data. Working with more than 250 archaeologists and anthropologists around the world, they generated new genetic sequences from 10,016 ancient individuals across West Eurasia, the region that today covers Europe and parts of the Middle East. They added those to roughly 5,800 previously published ancient genomes, bringing the total to nearly 16,000 ancient people. As Reich put it, this one paper doubles the size of the entire ancient human DNA record. Every skeleton in that collection is a single frame. Lined up in order across 10,000 years, they become something close to a film of our species changing. But the data alone would not have been enough. The breakthrough that made the study possible came from Ali Akbari, the first author and a senior scientist in Reich's lab. Akbari developed a new computational method designed to do the one thing nobody could do before: isolate the faint signal of true directional selection from all the noise of migration, mixing, and chance. Rather than starting with a model of how selection should look and hunting for matches, his approach let the patterns and the data reveal themselves. The distinction matters more than it sounds. An older method asks whether the data fit a particular story of selection and tends to find what it is looking for. Akbari's method instead tracks how each gene variant actually moves through time and flags only the changes too large and too consistent to be explained by chance or migration. It is the difference between assuming a verdict and weighing the evidence. In Akbari's words, the goal was to let the data speak for itself, so the team could watch selection shape human biology almost in real time. When he ran the analysis, the first finding was a lesson in humility. Across all the changes in gene frequency over 10,000 years, directional selection accounted for only about 2%. 98% of the change came from migration, mixing, and drift. Selection, it turned out, was a quiet force. Easy to miss, easy to overstate. But 2% of a genome this size is still an enormous amount of DNA. Within the 2%, Akbari pinpointed 479 specific gene variants that had been strongly pushed up or down in frequency by selection. Not 21, 479. The number of confirmed cases jumped by more than 20-fold, and the team flagged over 7,600 additional sites that have better than even odds of being real and now await follow-up. The single most striking result was about timing. Because the data spanned 10,000 years, Akbari could measure not just whether selection happened, but when it happened, and how fast. The rate was not steady. Selection accelerated after people abandoned hunting and gathering and took up farming. This makes a kind of grim sense. Agriculture rewrote the human environment. It packed people into denser settlements, changed the diet completely, brought them into constant contact with livestock and the diseases livestock carry, and concentrated populations where epidemics could spread. Bioarchaeologists have known for decades that the first farmers paid a steep price for their fields. Skeletons from early farming communities are often shorter than those of the hunter-gatherers before them. Their teeth are riddled with cavities from a starch-heavy diet. Their bones carry the marks of iron deficiency and chronic infection. Crowded together with their animals, they met tuberculosis, new strains of disease and infections that had never troubled small, mobile bands. A population under that much new stress is a population under selection. Every epidemic that swept a Neolithic village was, in genetic terms, a filter. And the genes that helped people survive it were passed on more often than the genes that did not. A new world creates new pressures, and new pressures favor new genes. Farming did not free us from natural selection. It handed selection a longer list of things to work on. So what exactly did it work on? Some of the selected variants are the familiar markers of European ancestry. Genes for lighter skin and for red hair both show the fingerprints of strong selection. The story of skin color is more recent than most people assume. The hunter-gatherers who lived in Europe after the Ice Age most likely had dark or intermediate skin, and the gene variants behind pale European skin became common only over the last several thousand years, carried in part by incoming farmers and then pushed higher by selection. The leading explanation is sunlight. Farther from the equator, under weaker sun and on a farming diet lower in vitamin D than a hunter's, lighter skin let the body make more of that vitamin from limited light, and selection rewarded it. Other variants are tied directly to the immune system, which is no surprise once you remember what farming brought with it. The team found selection on variants linked to resistance to leprosy, and even on a variant that today is associated with immunity to HIV infection, a virus that did not exist in the ancient world. which tells you the gene was originally doing something else entirely. There was selection against male pattern baldness, against rheumatoid arthritis, and against a predisposition to alcoholism. The B version of the gene behind blood type, which influences how the body resists certain bacteria and viruses, also rose under selection. Run down the full list and a pattern emerges. A striking share of the strongly selected genes touch the immune system in one way or another. That is the signature of a species locked in a long war with infection. For the 10,000 years this study covers, the deadliest force shaping the human genome was almost certainly disease. Many of the most interesting signals were not single genes at all. For some traits, dozens of variants scattered across the genome were nudged in the same direction at once, each contributing a little. This is polygenic selection, and it is far harder to detect than a single dramatic gene, because no individual variant changes very much. Catching it required exactly the scale of data this study assembled. The list of affected traits is long and often surprising. Selection favored a faster natural walking pace, which is a marker today of general health and longevity. It pushed down body fat, waist-to-hip ratio, and overall body mass. It even acted on variants linked today to a lower susceptibility to tobacco smoking, thousands of years before tobacco ever reached the region. Another reminder that a gene's modern label rarely matches its ancient job. Then there are the findings that seem to contradict themselves. The clearest example involves gluten. The major genetic risk factor for celiac disease, an autoimmune reaction to gluten, actually became more common right after people began farming wheat. At first glance, that is backwards. Why would selection favor a gene that makes you intolerant of the very crop you now depend on? The answer the researchers offer is a warning about how to read this data at all. A gene variant rarely does just one thing. The variant that raises celiac risk almost certainly carried some other advantage in the ancient world, perhaps a sharper immune response against a deadly pathogen, and the gluten sensitivity simply came along for the ride. Whatever it cost in digestion, it paid for in survival. This is the point in the study where careful science separates itself from a tempting headline, and it is the point the researchers stress most firmly. Among the 479 variants, more than 60% are linked to traits we can measure in people today. Some of those traits sound provocative. The flagged variants include ones associated in modern databases with cognitive test scores, with years of schooling, even with household income. It would be easy, and completely wrong, to read that as proof that Europeans were selected to become smarter or wealthier. The authors shut the interpretation down directly. Consider what a database actually records. It notes that people carrying a certain variant today tend, on average, to stay in school slightly longer. But staying in school is a product of the modern world, of literacy, institutions, and economies that did not exist when the variant was rising 8,000 years ago. Whatever that gene was doing then, it was not helping anyone pass an exam. What a variant is associated with now is not the same as why it spread then. The honest conclusion is narrow but solid. Selection touched these variants in the past, and only molecular biology, not a modern survey, can eventually tell us why. There are two more traps the team built their method to avoid, and both reveal how cautious good genetics has to be. The first is that a flagged variant might not be the real target at all. Genes sit close together on a chromosome, and a neighbor can be dragged along when selection grips the gene beside it, another case of coming along for the ride. The second is that the meaning of a variant changes with the environment. Some of the variants tied today to tuberculosis or multiple sclerosis did not simply rise. They rose and then fell across the millennia, as the pressures around them shifted, as if natural selection changed its mind. A trait that protected your great-great-grandparents could become a liability in their descendants. The classic illustration, which the researchers point to, is the sickle cell variant, which causes disease in one form but shields against malaria in another. What looks like selection for a sickness can really be selection against a different one. What the study delivers, then, is not a tidy story about why we look the way we do. It is something more rigorous and more honest. The first detailed map of when and where selection acted on the human genome, built from real bodies rather than from assumptions. Reich describes it as the ability to assign a place and a time to the forces that shaped us. The team has made all of its data and methods freely available so other researchers can test them. That openness matters because the 479 confirmed variants are not the end of the list. Thousands of weaker candidates flagged by the same analysis are now waiting for other labs to confirm or rule out. And the obvious next question is already being asked. West Eurasia is only one corner of humanity. Reich wants to know whether the same core traits were under selection in East Asia, in East Africa, and among the first peoples of the Americas. Whether evolution wrote similar chapters across the continents or completely different ones. The findings reach beyond ancient history. Akbari points out a practical consequence for modern medicine. If scientists are designing a gene therapy that switches off a particular variant, it would be worth checking whether that variant was strongly favored by natural selection in the past. A gene our ancestors were selected to keep may be doing something important that we do not yet understand, and switching it off could carry a hidden cost. There is a flip side that could matter even more. Some of the variants natural selection favored in the past are the same ones that raise the risk of disease today, in conditions ranging from diabetes to inflammatory and autoimmune disorders. A gene that protected an early farmer from a killer infection can, in a modern body with a modern diet and a far longer lifespan, turn into a liability. Understanding that trade-off, what these genes bought our ancestors and what they now cost us, is one of the clearest practical reasons to read the deep history of our own DNA. 10,000 years of human survival is, in effect, a long and expensive experiment whose results are written in our cells, and we are only now learning to read them. The deepest implication is the simplest one. We did not finish evolving when we left the savannah, or when we planted the first fields, or when we built the first cities. The data show selection still reshaping our species well into recorded history, in the genes of people whose descendants are alive today. Whatever pressures our modern world is now applying, its diets, its diseases, its crowded cities, are almost certainly leaving their own marks in genomes that future scientists will one day read. The film of human evolution did not end. We are still in it.